Okay, this time for sure. We're finishing this chapter in this video. So I've gone a little over. These are kind of challenging concepts, so do take your time. Please come with any questions you have about this when you see me, or email me with questions. So, of course, this whole process of producing new cells through mitosis, right, dividing, has to be carefully controlled because, right, the idea of developing a tumor is simply the cell cycle's out of control. You can't stop it. And, of course, if tumor cells spread, we call that cancer. And on the opposite side, speaking of homeostasis, right, so there's a balance in cell number. On the opposite side, if you don't produce enough cells, in other words, undergo mitosis often enough, you basically put yourself at risk for ulceration, right? And then ulceration can lead to secondary infections, which can kill people and often do. All right, so here, and this I'm not going to test you on, but I think it's something to consider, right? Here's some. I mentioned there's these internal checks and external checks. And there's specific places during that entire cell cycle in which the cell literally is not sentient, it's not stopping itself to think, but where there's a pause in the process, and to move on there has to be certain chemical factors available in certain situations available, and we call those, right, checkpoints. So there is definitely a balance in cell number, and if there wasn't, we said we have excessive would be tumors, cancer. Not enough would be ulceration. So these cells are programmed. Yes, this is in their genes. When we say program, we mean that there's a genetic code. So cells containing in their genes actually have the ability to make chemicals that helps the cell decide. So internal and external factors. So something from the outside, like a hormone, as I said, growth hormone, or something from the inside, like a protein known as cyclin B or CDK. So I say here again, and anything in italics I'm not going to ask you about, but checkpoints are basically quality control. Cyclin and cyclin-dependent kinases, that's what CDK stands for, are important proteins to help promote cell division. So if they are not around, then the cell stops and won't divide. Okay, so here's external and internal. Again, remember, got to control this. How often do we undergo division? When do we divide? So hormones that stimulate mitosis, such as things like growth hormones, right, are very important to this process. There are a number of steroids like testosterone and estrogen that also stimulate mitosis. Growth factors. So these are things that one cell type will make and release, and it affects another cell. And we have a whole bunch of different growth factors. And they stimulate mitosis, as the name implies. But they tend to be more in a certain area, whereas hormones can be released and they can have a wide effect all over the body. And as I said previously in the previous video, I said, how much space you have. Because once cells make contact with another cell, they stop dividing. They are not going to overcrowd and run out of resources and become disorganized. And we call that contact inhibition. So as soon as we've used up all the space, you're not going to divide anymore. You go into a senescence, as I seem to have right now. Internal factors. So within the cell, there are these things called telomeres, and this is code on the end of the chromosomes, and it's kind of an indicator for how often can you divide. There's levels of proteins that have to be there, a certain amount of these, to allow the cell to divide, and we gave you in the previous slide kinases and cyclins, and of course cell size. So the bigger a cell grows, the more likely it is to divide, because if you get too large, you cannot move your molecules efficiently, and you'll die. All right. So here's the loss of control of cell cycle and tumors. As I said, here's images of cancerous cells and the normal cells, and cancerous cells generally lose their organization. So there's indicators, morphology and genetics you can use right, and we do when we biopsy. So a tumor, again, is any cell is the result of a cell that's lost its control of cell cycle, and it just keeps dividing. All right, they can be benign, which means they stay right where they are, where they became abnormal, and they just stay there and stay contained in the body. or they can be malignant. And when they're malignant, we call them cancer, and we use this term metastasize. So basic characteristics, right? You lose control of your cell cycle, and one cancer cell, again, will pass on this defective code to another. It is, it says, here transplantability, but we're not going to worry about that right now. They don't have a specific function. This is really the key issue here. Is these cells, see how they don't look alike anymore? Well, they also don't do the same thing that these cells did. So now they can crowd out these cells, overgrow them, and overwhelm them. And as they do, these cells, right, are going to die off and lose function. The whole organ's going to lose function. They have an ability to cause blood cells to grow to them, called angiogenesis. They are invasive, and this term metastasizes is part of that too, but invasive means they can grow into a blood vessel or limb. What kinds of cells are most cancers? Epithelial and then connective, because epithelial cells divide all throughout your life. You're not often going to see a neural or a muscle cell origin for cancers. So it does happen, but it's not as common as epithelial cells. So I said, in case you missed this on the last slide, cancers do require genetic changes. So the cell needs to be told when to divide and when to stop, and genes help with this. So two groups of genes involved in cancer are going to be the genes that tell them to stop and start. And these are known as the tumor suppressor genes. This is a whole family of genes that says stop. And they are part of these checkpoints as well. And then these proto-oncogenes are a group of genes that stimulate cells to divide and tell them to go forth. And you have to have a balance between the two, because you have to start and you have to stop. All right? So proto-oncogenes, and this is what I want you to know for me, is that they increase cell division. But they can be mutated, right? Because cancers are the result of genetic changes, and genetic changes are called mutations. So when we mutate a proto-oncogene, we then call it an oncogene, and there you get the name oncology, right? And these cells keep on dividing. It's like removing the brakes. Tumor suppressor genes, a whole nother family of genes, stop or slow down mitosis. So that's what they do normally. But if you've mutated the brakes, well then you increase cell division. And I say here that environmental factors are what can often lead to the changes in these two groups. And I'll give you examples like toxic chemicals, radiation, drugs. All right? So we know how to get 1 trillion cells. Divide, 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 divide. How do we get 290 different cell types? And we know now that there's far more than that. So this last concept is about stem and progenitor cells. So where we started, right? Where we first came in. Yay, we came back to where we started, right? The penultimate, stem cell that we call totipotent, because it has total potential, was that zygote, right? Because everything came from that. And as a stem gives rise to flowers and buds and, leaves and stem and more branches, hence a stem cell is able to give rise to lots of different structures. And we actually mean cells that become structures. And then progenitor cells are the cells that come from these stem cells. So I, just playfully ask, do you know what you're going to do when you grow up? Well, a stem cell doesn't. All right, it hasn't defined that. So we often say that it is not differentiated. It doesn't know the difference. So we describe these by their potential to produce new cell types. So we use the term totipotent, pluripotent, multipotent, and I say, there's even oligopotent and unipotent, but you don't have to know those. All right? So the stem cell are cells that retain this ability to divide repeatedly. So they undergo division faster, typically, and they produce cells that are unspecialized. Again, they aren't grown up, basically aren't differentiated. And that's good, because then they can become a variety of different cells. All right? Progenitors then are the daughter cells, which have some specialization, but are not fully differentiated. In other words, they aren't really sure what they're going to be, but I'm going to be something here in the healthcare field, right? Maybe a nurse, a doctor, a radiologist, but I've kind of narrowed down my options, all right, when you're a progenitor. All right, so here's a nice cartoon. Ooh, that is really kind of, sorry about that. Didn't realize that was so fuzzy here, or pixelated. But here, we're looking at this totipotent embryonic stem cell, and it's your zygote, right? And you divide, and you start to produce, over about a 2-week period, these 3 types of different tissues, but tissues made of cells. So these are now pluripotent. So they can make a lot of different things, but not everything. So here's our endoderm, our mesoderm, and our ectoderm. Turns out the ectoderm is going to give rise to your nervous system, your integument. Your endoderm is going to be linings of all the stuff inside of you, and mesoderm is the middle stuff, the muscle, the bone, the blood cells, etc. All right? So more time goes by, right? And these start to become more determined in what they're going to be. They produce a whole lot more themselves, and then we say, they're multipotent stem cells, and they would give rise to then specific, right? So examples of these, as they say here, adult bone marrow. So adult bone marrow is a multipotent, and it is because that hematocytoblast that you'll look at in AMP2, right, gives rise to all the blood types, all the blood cells, excuse me, not types, all the blood cells, right? Erythrocytes, red blood cells, the 5 types of white blood cells, and platelets all come from this singular, right, adult bone marrow cell. So it's a multipotent stem cell, right? So when differentiation occurs, it means there's a change in the size, shape, metabolic activity, and function. And this happens because genes, right, get turned on and off. We change our gene expression. All right? So there you have it. I hope you are thoroughly not confused. No, I hope that some of it's making sense. Please again remember that I'm not going to hold you responsible for some of those specifics in this last video, but it is important to get the broad concepts of how this imbalance in cell cycle, and be able to define stem cells and progenitor cells, etc. So thanks for watching.